Hello, everybody. Welcome today. My name is Justin Dela Cruz. I'm with the network of the National Library of Medicine, National Center for Data Services. Wanted to welcome you today to this session, Greatest Crimes and Statistics, with Marika Jones, who is a senior biostatistician at University of Virginia. We're very happy to have her join us today and talk about statistics, which will help round out some of the things we've been doing with data science. So if you want to see any of our other offerings, check out our website. I'll put that link in the chat. But for now, I'll hand it over to Marika.
Thank you, Justin. Thank you to everybody who joined, and thanks for popping into the chat and telling us what corner of the world you're from. That is so fun to see everybody joining in from everywhere. So keep it coming. There will be plenty of opportunities to contribute in the chat. And also, if you have a question for me, pop it into the chat and Justin will monitor. There will be a few opportunities to take questions about the given topics as we go through. All right, so today we're going to talk about the greatest crimes in statistics, and I hope that I can give you guys some actionable things to actually do as you're reading literature and thinking about research to improve your understanding of statistics and your critical eye or critical brain when it comes to reading journal articles. So the reason why this is near and dear to my heart is that an estimate says 75 to 90% of preclinical results can't be replicated. And that's pretty bad. We're kind of facing a bit of a scientific crisis right now, where people are starting to mistrust science and scientists, and it's not getting much better in the future. So one of those places where I think we can do a little bit better is education and statistics. So that's why I'm here. That lack of replication equates to $28 billion a year, which is enough for every person on the globe to get a fancy latte, not just a drip coffee, fancy latte from Starbucks. And that's in the US alone. So when you multiply that out across the world, it's really quite a big waste of money and resources to chasing the truth when we're not doing such a great job at it. So we're beginning to look a bit like snake oil salesmen, and we're facing a real credibility crisis. So I'm hopeful that I can make a small, tiny, tiny dent in that problem by offering up a little bit of education. So what are the problems? One study in 2016 polled scientists, and scientists themselves said that statistics was one of the top reasons why they were not doing such great science themselves. So if I can contribute in any tiny way to that, that is what I'm here to do. So your turn. At your institution, how do researchers learn to carry out the correct statistical analyses? And what do they do when they need help? Where do they go, or who can they talk to at your institution? Go ahead and write it in the chat. Cool. I see grad students, data center, biostatisticians, the library, word of mouth, informal mentors, a research department, a CTSI, public health school, GIS and data center in the library. Yeah, excellent. They're PIs, right? Online resources, thank you. Excellent. I wish that there were a more robust resource network around universities and research centers around the country where people could get more help than they currently do. A lot of it is informal. With the students that I talk to, my statistics core can only serve so many students, and of course we are for cost. Libraries are wonderful in that they are not usually, that you guys can help people for free often, which is so, so wonderful. But yeah, we really do need a bit more robust resources, support network around statistics, in my opinion. So today we're going to tackle 4 different topics: data visualization, p-hacking, how to report your results, and pseudoreplication. There's so much more to cover here in statistics that 1 hour doesn't quite cut it. So if you're interested in a deeper dive, I'll be offering a self-paced NNLM course called Understand the Methods Section, an Introduction to Statistics, in the near future. So check on NNLM's website for more as that gets developed. All right, so first up is data viz. My tips on data viz are show the data as best you can and maximize the information to ink ratio, meaning remove pieces of the plot that you don't really need. So we'll talk about when and why you might need certain pieces. All right, we're going to play optometrist. I'm going to open up the chat. We'll call number 1 on the left and number 2 on the right. Which plot is better, 1 or 2, and why? I don't think the optometrist asks you why. Oh, we have pretty equal votes between 1 and 2. I love it when we're split. Cool, and many people are offering their contributions on their reasons. Number 1 is really, really nice because it's so simple. Everybody can get it. The story is very clear. The control is higher than the treatment. And there's not much to complain about in there, right? But it may be hiding a lot behind the scenes. So number 2 on the right-hand side has a lot more information. Number 2 is called a strip chart plus summary statistic. That's kind of a mouthful, right? The strip chart portion is the piece with all the dots. And so you can see every single data point, and you can kind of tell the treatment group is actually bimodal. There's 2 groups within the treatment group, a group of 4 that are kind of higher and a group of 4 that are kind of lower. And then in the control group, we have a wide spread. We see high variance there, and even one kind of outlying point on the bottom. That's a lot more information than we were able to get with the bar chart on the left. So pros and cons for each, depending on what you want to do. It will depend on what kind of visualization you create. Typically speaking, for scientific publications, the one on the right will be preferred because it's a lot more transparent, tells people a lot more about what's going on in these 2 groups. Maybe you have discussion points about that outlier or about the bimodal distribution, whereas on the left, you're taking up a lot of real estate in a manuscript that doesn't have a lot of real estate to give with one very basic graphic that basically shows 2 different numbers with a lot of space, using a lot of space. All right, another opportunity for you guys to engage. Here is a plot of votes cast in New Zealand by 4 different political parties. So political parties are along the x-axis and the number of votes are along the y, the vertical. What things do you not like about this plot that you wish you could improve? Get rid of the 3D. Yes, Nancy, agreed. And Hannah, excellent. Sort them instead of alphabetical. Yes, good eye. These parties are alphabetical, but probably that's not the best option here. We can't see a big difference between the tiny ones. Yes. There's too many groups. Yeah, definitely. It needs to be sorted. Excellent. Exact number, yeah, maybe some annotations would help. Some color, yes, the color between the data and the background is not very distinct. We have like a light blue and a light gray. Excellent. So here's a redo on the next slide where we flipped the graph. It used to be in many statistical or graphing softwares that you could only put data with letters like these political parties on the X, but that is not the case anymore. Many programs like Excel or R or SPSS will allow you to put a categorical variable along the Y. So please don't feel trapped. Once you put it on the Y, now all of a sudden you can read them properly. You don't have to tilt your head to read them. We've also added annotations, really good distinction between the data and the background. So that's part of our principle of show the data. And now we're able to tell the difference between the smallest groups down at the bottom of this plot because of those annotations. So a much more successful graph here. And I hope that you guys can take these kind of lessons and apply them to plots that you make for presentations, or if you're helping researchers make plots for their presentations or their manuscripts, keep these principles in mind to show the data, meaning that the data should stand out from the background, Perhaps also that means annotations, and sort intelligently. Here's another example of sorting. On the left-hand side, we have the countries sorted alphabetically, which is the default in most programs. But as you can tell, we can't really tell a story out of this left-hand side. It's pretty meaningless, kind of jumbled. And then as soon as we sort them, now we have a story. The United States spends a bunch more money on healthcare spending as a percentage of GDP than most other countries. Check out that gap between the US and the next countries. That is wild. As opposed to on the left-hand side, we really could not tell that at all. The United States is hidden down at the bottom, and our eye is not drawn to it at all. Question I get asked often is, do I need to plot with zero? On the left-hand side here are bars. So the encoding of the data is in a bar chart where the important information is the length of the bar from 0 up to its number. And we have A at 96, B at 98, and C at 99. They don't look very different to the eye at all. So that tells quite a different story than on the right-hand side, where we have probably a mean and a standard deviation or a mean and a standard error. They're not labeled, so we don't really know what those error bars are, but they look really different. The error bars do not overlap. And now I'm seeing a kind of different story that A is much lower than B, which is much lower than C on the right. So my advice is, depending on your story, that should dictate what kind of plot you develop. If there is no difference between these statistically, it might be nicer to show people the plot on the left. The plot on the left is lacking error bars, so I would probably advocate for error bars over there. But you can get away with not showing 0 on the right-hand side because the encoding is really the position along the y-axis of these horizontal lines, rather than the length of the bar from 0. So depending on your encoding, you may or may not have to include 0. For a bar plot, yes, you do need 0, but you could easily change this bar plot into a different encoding, like this summary statistic plot on the right. This graphic is showing a bar plot on the left, which is kind of the traditional way of showing this kind of data, where you have two groups and you want to show a difference between the two groups. Unfortunately, the bar plot could be hiding a bunch of things underneath it. So this article from Weisgerber is advocating for no more bar plots unless you have a bunch of data, and showing every single data point would be overwhelming. And even then I would probably advocate to show every data point and have a summary statistic on top. So summary statistics are really only meaningful when there are enough data to summarize. We think by eye that what's hiding underneath the plot in A looks beautiful, like in B. But really, it may not be so beautiful. Like in C, we have an outlier in the second group. In D, both groups have bimodal distributions. And in E, there were really unequal ends. Each of those situations come with different statistics. So down at the bottom, we have these t-tests that are comparing these two groups. And sometimes that t-test turns out to be significant, and sometimes it doesn't, depending on what's hiding underneath the bar plot. So a bar plot can obscure a lot of different information that could be hiding underneath it, which is why we're advocating for showing the actual data when possible. Can you do a bar plot with an overlay of the actual data? Of course, yes. The whole world is open to you. You can do whatever you like, but it does have consequences in what people are concluding about your data. Similarly, in A, if these were paired values, we are hoping that what's underneath there looks like in B, where everybody kind of had an average increase, and the increase that everybody saw is sort of the same as the difference between those two bars in A. But really what could be happening is more like in C or in D, where some people had a drastic increase and other people declined or stayed stagnant, Maybe this is a treatment and it didn't really help some people, and it really drastically helped some other people. So the story could be a bit more messy than what we assume is hiding underneath A. So if you have paired data, I would encourage you to make a paired plot to show those data. All right, I'm going to stop here for just a second and check in with Justin about questions. If we had any questions in the chat, let me know, Justin.
I haven't seen any outside of the large number of responses that came in through. Does anyone have any questions at the moment?
Pop them in the chat if you have a question about plots.
Do you have to have special software for showing all the different data points?
Excel will do it. Excel is actually not a bad graphing software in my opinion. Sometimes it's a bit clunky to get to the actual settings that you want, but you can definitely show all of the data points with Excel. Other software like common graphing software in my world is R. I am fully in R. Everything I do is in R. And of course that can do it. Other software that my colleagues use is Prism. Prism is more for our basic scientists, but people rave about their plotting abilities.
Did you say that you do have to start at 0 with bar graphs, but if you can change to a different plot type, then you don't have to start at 0?
That's right. A bar encodes the data as the distance from 0 to the number. So it's the length of the bar that is the important component of a bar plot. Whereas the other chart that we were looking at was more like a mini box plot where it just had a horizontal line for the mean and then horizontal lines for the mean plus and minus standard deviation. And those are just read off of the y-axis. So for those, the encoding is the position along the y. So yes, I would say for a bar plot, you must show the 0 line. And for other types of encodings, it may be more optional.
All right. That's all I see for now.
Thank you guys so much. Keep them coming, okay? All right, next up is about P. What do we know about P? Most people know that 0.05 is some kind of magic number, and we're at 0.05, and at 0.05, something really special happens. But the real definition of the p-value is a lot more important than the actual like decision point that people use. P is really the probability of observing what you observed or something more extreme if the null hypothesis is true. So given that nothing cool is happening, that just chance is occurring, how often would I see the result that I saw? Is the result that I saw really uncommon? Should I be very surprised? If I'm very surprised, I would probably throw out my null hypothesis. It's not really probable that nothing's happening. So something else must be occurring. Whereas if the P is very big, I'm not that surprised. It definitely could have occurred if the null hypothesis is true. And so we keep the null hypothesis. Now this dichotomy of P bigger than 0.05, nothing cool, go back to the drawing board, versus P less than 0.05, you're super cool, you can go ahead and publish your results. That's problematic in my opinion. So we're going to talk about some strategies of what we can do to kind of fix that or to move towards fixing that in our lives. Getting rid of p-values, I think, is impractical, right? They are the currency of the scientific world right now. So we're somewhat stuck with them. We're not going to change the cutoff either, but maybe we can work towards a little bit more thoughtfulness around p-values. Has anybody heard of p-hacking? Pop something in the chat if you have heard of p-hacking. What is it? Yes, we've heard of it. That's great. No, we've heard of it. Okay. No, we have not heard of it. Any ideas for those of you who have heard of it? Cheating-ish. Yeah, I like that. Excellent, Jocelyn. Keep looking until you find a significant p-value. Exactly. Digging around. Yes. Adjusting stats to fit your hypothesis. Yes. Manipulating the data. Yes. Manipulating the statistics. Yes. Everything about that is p-hacking, all of those. So here's some ideas. that I have seen in my real life. Stop collecting data once the p is less than 0.05. Many students will come to me and say, my p is not less than 0.05. I'm going to go upstairs and collect some more mice. No, don't do that. Analyze many outcomes, but only report those that worked, right? I've seen that before. Collect and analyze many conditions. So maybe you have 10-week-old mice and 50-week-old mice and whatever other age, and your manipulation only worked in the youngest of mice. So you only write about those in your paper. That's also p-hacking. Add covariates. These are extra x's, extra variables that you wanted to analyze until your p is less than 0.05. Exclude participants, maybe outliers, until your p is less than 0.05. Add some more sample size until your p is less than 0.05, or maybe a transformation of the data that can get you to a p of 0.05. So we're always chasing this p of less than 0.05. That's problematic, but many of these are actually important. Sometimes you do want to remove outliers. Sometimes you do need to transform the data. So my question, how do you know that you or a researcher is doing the best data analysis and not just p-hacking? And how do you get enough flexibility to add a transformation if needed that makes your analysis more correct when I just said that that was p-hacking? So what makes an analysis decision acceptable and what makes it p-hacking? How can we fix it? Yeah, you librarians know. Pre-specify your protocol. Absolutely. Do grad students do that? Hardly ever, right? So what is this pre-specifying or registering a protocol? It would be at its minimum writing down what you plan to do. Do you plan to collect 10 mice or 100 mice or, 10 humans or 100 humans? Do you plan to transform your data? In what way? Why? What are the decision thresholds around there? And that's really difficult, right? You probably need a bit of experience with this type of data, whatever you're about to do. So it might be nice to speak with senior members of your lab or with people who have used this type of data before and understand what kinds of transformations are typical. What kind of sample size is appropriate? Perhaps it would be nice to do a power calculation or a sample size analysis ahead of time so that you know how many people to include in your study a priori, rather than deciding on the fly based on the p-value. Ideally, you would like to draft your data analysis plan before you even go and collect your data so that you're ready to make the proper decisions. And if something should change on the way, like I just had a study where something changed and our data did not meet the assumptions that we thought it would, we register a change in the protocol. You write down the data distribution looks a little different than I thought. There's actually zero inflation when we weren't expecting that. And as a result, we plan to analyze it like this instead. Okay, so registering your decisions as you make them to make sure that the decisions are made in honor of the data rather than in chasing a P. Anytime you're making a decision to chase a P, that's probably not such great science, but we definitely need the ability to change our data analysis plans in case something happens that we weren't expecting. I would like to see a lot of work on this at the university or institution level in the future. I think researchers can do a much better job coaching graduate students to write some of this stuff down ahead of time so that we're not just chasing the data all over the place. Make a plan ahead of time. And students are woefully in the dark about how to do that. So a lot more education needed there. All right, we'll stop here and take questions about p-values or p-hacking. If you have any questions about p-values, p-hacking, pop them in the chat.
There was a comment earlier that got some agreements. Some of this could be addressed if journals were more willing to accept articles that didn't show an effect or even a negative effect.
Yes, absolutely. Changes needed at all levels, including the journal level, institution level, researcher level.
Do you have any examples of famous studies that are p-hacked?
That is a great question. I don't off the top of my head, but I'm about to go home and Google that. I don't know if it would come out. I mean, maybe if there was like a failure to replicate and then you found out that they actually made a decision in chasing a p-value. I'm not sure if we would know if they p-hacked because it's in how they make their decisions. There's a ton of studies that can't be replicated.
Is it common to explain in your article why you chose the particular statistical test that you used?
To explain in your methods?
Yeah, is it common for researchers to write it out in their article why they chose the statistical test that they chose?
Yeah, I would say that's fairly common to say my data were zero inflated and thus we chose a zero inflated Poisson model, something like that. That's rationale for why. Data were not normal and so a log transform was used. That's pretty common.
Besides protocol and protocol registration, are there other lines of discernment of acceptable changes versus cheating in data analysis?
I would say that most fields are not quite there yet with pre-registration, like formal pre-registration. It's pretty uncommon still in most of biomedical literature and preclinical type stuff, unless you're doing like a registered clinical trial. So in those kind of fields, I think it's up to the PI to really say like, on Tuesday, we're going to go through your statistical analysis plan, so please write something down and then to really help the student through that statistical analysis plan and writing down decisions that had to change as they go. So a much more informal process, I think, would work and should be implemented more often.
Could you mention what it means to register a protocol? Is that just doing it within your lab work or lab notes, or is there some other process?
There are formalized processes for cancer trials and for RCTs, but as I said, many fields do not have a formal process. The formal process is sometimes through a journal, where if you pre-register your protocol, the journal can say, we like your methods and we think that this is worthwhile. Go ahead and do your research and we will publish whatever you find. So that sort of takes away the problem of not publishing negative results, where the journal pledges to publish no matter what you find based on your methods, which is kind of a cool idea.
Well, that is cool. Those are all the questions I saw for now.
Those are great, you guys. Thank you so much. Next up is about how to report your results, and it's kind of related to p-hacking. So oftentimes we put so much stock in p-values that we kind of forget what p-values are and how they're influenced. So here's two quotes. Statistical significance is the least interesting thing about the results. You should describe the results in terms of the measures of magnitude, not just does the treatment affect people, but how much does it affect them, by the statistician Jean Glass. And the primary product of our research inquiry is one or more measures of effect size, not p-values, by Jacob Cohen, another famous statistician. So up at the top, I have a little image of heart attacks and bear taking aspirin. And this is a study about how bear aspirin can prevent heart attacks, and that p-value is so super significant. P is less than 0.00001. That looks like it's really important. Maybe we should all take aspirin. But actually, if you look at the surrounding statistics, like the sample size was 22,000 people. The risk difference was 0.77%. So we're actually not preventing that many heart attacks by putting everybody on bear. I am not recommending putting everybody on aspirin. And the R squared, which is another measure of model fit or of effect size, is also very, very, very small, suggesting that taking aspirin is only responsible for 0.1% of heart attacks, of the variability in heart attacks. That's not that much. So once you look at the surrounding statistics, that P of less than 0.00001 is no longer super compelling. So that's my point here on this slide with these quotes, is that P needs to be supported by other friends and other statistics and should not be just presented naked. So what is P and what is effect size? We just covered P. P is the probability that your result is due to chance, or that you would see this result under the null hypothesis. And when P is less than 0.05, it's typically considered significant. And significant means that it was not due to chance, that something else is happening. Maybe there is a difference in the groups, something like that. The effect size is more about the magnitude of the result. It could be a difference in the groups if that's your kind of like study design. If there's two groups, you want to know the difference between the groups. The effect size would be something like the T statistic that tells you the difference between the groups. If that's not your study design, maybe it's something like a risk reduction that we saw on the previous slide with the aspirin. Or maybe it's something like a model fit, or how much the model fit changes if you remove one of your variables, something like that. So effect size is kind of vague, and maybe you haven't gotten a good definition of it because it depends on what kind of study you're doing. So we can think about it as the magnitude of the result, and it can be defined differently for different studies, but maybe the best one to just kind of lodge in your brain is about the difference between the groups. So I went online and found tables. And this table I thought was particularly indicative of naked p-values. We have these variables along the left-hand side, and for each variable, we just get to know whether it was favorable or unfavorable. I don't even know what that means. And then a p-value, and that's it. It doesn't really tell us about the magnitude of the effect. How much does gender impact whatever they were studying? We have no idea. Okay, and I didn't put the citation because I don't want to call these people out too harshly. So this is just an example of a scientific table that has naked p-values, so you can help your researchers avoid that. P should always have some kind of support with it. So how to do this better? We would like, ideally, for you to pair your statistics with some measure of uncertainty and then also include the p-value because, as I said, that is the current currency of scientific research. So here's a table with some groups along the top and then some variables along the left-hand side. We get to know about the mean, plus and minus the standard error of the mean, so some measure of variability. And then in the asterisks is where we can see our p-values. And they do stand out visually. I understand why people say p-values need to be in the table, but here I would argue that they are quite visually apparent with those blue asterisks. Okay, so this table I think is a good example of showing the proper statistics. Our eyes are drawn to the difference in the means. So for HW, the first variable, we can tell that the middle group is way lower than the other two groups. Just looking at the mean, it's visually apparent, and we're given some kind of magnitude of the effect. So I think this is a successful table. A different kind of study might have something else, like an odds ratio reported. That is also a measure of effect size, plus its measure of uncertainty, like a 95% confidence interval, something like that. And then a column for p-values, sure, with those that are significant bolded. Okay, great. But notice that we're including effect size and a measure of uncertainty as well. All right, we'll stop there for questions about how to report results.
Someone was asking about the difference between statistical significance and clinical significance.
Yeah, great question. I think a good example is the aspirin for heart attacks that we saw. It had great statistical significance, right? But if I told you that only 0.77% risk reduction, which is not going to affect that many people, now the clinical significance is kind of evaporating. So you do need some kind of knowledge of what the effect size would mean for people or mice or whatever you're studying. Like in the previous slide is the difference in HW between 97 and 120, 119. Is that important? I don't know that because I don't know what HW is and I don't know what these mice are. You know, like I'm not going to be able to tell you about clinical significance, but the clinician would. They would say, oh yeah, that's not that big a deal. We see that kind of difference all the time, something like that. So there's a huge difference between statistical significance and clinical significance. Statistical significance is just that p-value, less than 0.05, and clinical significance is what does it mean in the real world to have that magnitude of a difference. Stacy's asking to see the final table, maybe the odds ratio table. This one is a successful table because we have a column of effect size, which tells us about the magnitude of the effect. So we can see that the odds ratio is the highest in week 0 versus week 2. That's helpful. And for odds ratios, we may also want to look at the lowest because if you flip it, then that would be a high difference as well. And we're getting a 95% confidence interval there. So looking at the confidence interval tells us that, again, diet B week 0 compared to week 2 has the biggest difference. And then the p-values help us to evaluate the significance of those differences, showing that diet A, 0 to 2, 0 to 3, and 0 to 4 are statistically significant and the others are not. So I think this table is helpful because it has a measure of effect size plus the measure of uncertainty and also the p-value. The p-value is supported by other statistics. Excellent. We're going to dive into our last topic, and then at the end I have some discussion questions as well. Our last topic is about pseudoreplication. So pseudoreplication occurs, I think, because sample size is really tricky or can be tricky, especially for biology. So the hypotheses that we create, what we do experimentally, and what we measure may each be at a different level of the biological hierarchy. So on the bottom in this little schematic, I have a little hierarchy about atoms are smaller than molecules, which are smaller than macromolecules, organelles, cells, tissues, organs, systems, organisms, and then higher than that, populations, communities, ecosystems, and biospheres. So things are nested. So if we do a study, how do we know at which level we should determine the sample size? For example, let's say that you're studying mitochondria, and those mitochondria are inside cells, which are inside eyes, which are inside rats. So is it really the number of mitochondria that you study, or the number of cells, the number of eyes, or the number of rats? It can be kind of hard to tell. Obviously, if you measured a mouse, like from nose to tail, the measurement of the mouse would be the 1n. And if you measured that mouse 10 times and tried to say, no, 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 I have 10, people would all know that that's pseudoreplication. You cannot measure the same animal 10 times and call it an n of 10. However, things get a little bit more tricky once we're talking about a mouse that has, let's say, 2 kidneys, and then you take that kidney tissue and you slice it out onto slides. Let's say we have 7 slides, and then you're going to image the cells on those slides. Now you have thousands of cells, and maybe you're going to do your treatment on the cells on the slide. Now, because you did your treatment at a lower level, which one is the N? It gets a little tricky. So N is our sample size. That's going to be our definition. And N should be independently assigned to the experimental condition. If it's not, then that is considered pseudoreplication. So we're going to determine N at the level of the experimental condition. The problems with pseudoreplication are that if you have pseudoreplication, that's an overestimation of the sample size. Maybe you don't say you have 10 mice, you say you have, 100,000 cells. Now with that 100,000 cells, you're much more likely to say that there's a significant p-value. So sample size overestimation is correlated with an inflated statistical significance, which could lead to false positives. And our whole problem of doing science in not quite the best way and coming to false conclusions. There's also a problem that many statistical tests assume independence of your samples. And once your samples are no longer independent, you could get a false positive from that as well. So let's practice. This study is actually mine from my dissertation. I studied some wolves and I wanted to know if biomarker levels in the urine of these wolves was different depending on their reproductive status. So we have four groups. Wolves that are pregnant and gave birth to live young, wolves that are pregnant but the pups died, wolves that mated but did not become pregnant, and wolves that were housed singly and had no opportunity to mate. There were 13 wolves, 25 urine samples per wolf, and then each of those was run in triplicate. So that led to 961 urine samples. So in my manuscript, should I report 13, 25, or 961? What do you guys think and why? Were they all female? Yes, they were all female. Most people are saying 13, maybe 25. Is there a reason to state all of it? Yeah, maybe your manuscript is really explicit and you say all of it, but at some point you're going to need to know which numbers to actually put in your model. So yes, I advocate for being transparent and telling people all of this. 13 times 25, that's a good idea too. In this case, because the experimental manipulation is the group, this reproductive status group, then the N is applied to that level. So the sample size should be independently assigned to these reproductive groups. The thing that was independently assigned to the reproductive groups were the wolves. Each of the urine samples came from a wolf. So those are nested. Why were there so many samples per wolf? Yeah, good question. We were doing longitudinal studies. Yeah, the 25 are called technical replicates, or if your timing is also a variable, then maybe they're not all technical replicates, but we're getting into the weeds. The thing that was independently assigned to treatment group is wolf. And so in this study, wolves is your sample size. All of these other things are also important. And it's possible that in your model, you're actually going to put in all of those urine samples, all 961. So your models are going to be run on a sample size of 961. But hopefully you know that you'll need to run some kind of special model that accounts for the fact that the 961 are nested within those 25 urine samples, which are themselves nested within the wolf. So these data are clustered and need special treatment because of that. All right, one more practice. In this example, we have 10 mice. Then each of those 10 mice has 2 kidneys. And then we take out the slices of the kidney. Let's say we get 7 kidney sections. On the left-hand side, they're going to be treated with something. And on the right-hand side, they're going to be control sections. And then we image those sections, and we can count about 10,000 slides, 10,000 cells per slide. In this case, what do you think our sample size should be? 14 because it's 7 times 2? Yeah, that's good. The number of kidney sections? Yes, I think so. Potentially even 10. Yes, like Matt is saying. Yes, the experimental condition is applied to each of those tissue sections. So it's independently applied, sort of. But the independence is tricky, right? Because I'm not sure that those sections are actually independent. 7 sections come from one mouse. So probably, yeah, the kidney sections are not independent. Yes, Kristen, exactly right. And it's a paired study. Excellent, Nancy. Good. Because each of the kidneys is either treated or untreated from the same mouse. So we have some pairing going on and we have some nesting. So you could declare your N to be 7 per mouse, so 70 in the treated and 70 in the control. But I would argue that your statistical design best take into account that 7 sections came from the same kidney. That will be very, very important. So the levels of biological hierarchy are going to be critical here, and understanding some statistical techniques that can incorporate nesting is really, really important. So to that effect, we should use appropriate statistical analyses for the data that we have. Many, many times people are counseled to just average across the replicate measurements, and that loses a bunch of information. You didn't spend your whole day imaging 70 different kidney slices just to have one number, did you? No, you did that for a reason. And the reason is that there's variability within each of those slices and within each of those fields of view from the microscope and within each of those kidneys and within each of those mice. So we should honor the data that we collected and try to get a statistical model that incorporates the nesting structure of our data. If you have complex designs, an ANOVA and a t-test don't quite cut it. Even if you did average, I mean, it's just not a good idea to average, and then the t-test is just going to ignore all of the variability within those. So seek statistical support from your colleagues for these more complex designs. In case you're actually really interested in how we do this, there are more appropriate techniques for clustered or longitudinal data. Things like mixed effects models, which are abbreviated LMM or LME, generalized mixed effects models, which would be the GLMM, or something called generalized estimating equations, GEE, which is more for longitudinal data or for clustered data. So if you cared to go deep in there, those are the types of techniques that we would use for clustered or longitudinal data. So if you have that kind of data, think very, very carefully about how to analyze it rather than using just an ANOVA or a t-test. All right, there's a lovely paper that I'll close with from 2016 from the American Statistical Association with their recommendations for the future. Their recommendations follow an acronym, ATOMIC, which is kind of cool. The first one is to accept uncertainty, and part of accepting uncertainty is to write about it. So pair your point estimate like a mean with an uncertainty interval, like a standard deviation or a standard error, or pair your point estimate like an odds ratio with a 95% confidence interval, something like that. Be thoughtful. Often the methods are much more important than the p-value that was retrieved because of a lot of those things like p-hacking. So read the methods section and see if you think that they designed their study well, and if they did, then read on. Be open. I know librarians are very passionate about this. Share your data, your code, and your analyses to the best extent possible. Pre-register your studies to keep yourself honest. And remember that P is a continuous value rather than dichotomous. It really doesn't have a sharp cutoff, and it was not invented with the idea of this sharp cutoff. We just kind of invented that on the way. Be modest about the role of statistics in science and encourage replication attempts, and shoot for institutional change. What types of institutional change do we think are most important? Let's write about it in the chat. Yeah, great paper if you want to read the paper. There's a lot more in there. Yeah, journals publishing non-significant results. Absolutely. Collaboration and team science. Transparency and ethics. Absolutely. Journals. Yes. Readily available statistical consultants for free. Right? Yes, I would like that too, Joy. Excellent. More replication studies. Yeah, and funding for replication studies, right? It's not glorious, it's not glamorous, but it's really, really important. Less pressure to publish. Yes, for professors. Researchers with experience in the field need to actually lead. You can't expect the students to do the work if the mentors aren't helping. Yeah, agreed, sadly. More robust peer review. Yeah, this whole system is kind of rigged, right? Peer review, the peers who are reviewing are not compensated in any way. It's a huge time suck to review papers and really quite a challenge to offer a fair critical review. Yeah. Excellent. Thank you guys for participating so much. And with that, I'll put up my little credits.